Hi students, welcome to the developmental zoology class. Now let us going to see the topic is human cloning. Okay. First let us know about the cloning. What is cloning? The definition of the cloning is it is the production of the animals of the same type. Okay, same type of the animals are produced from the particular animal. Okay, that is cloning. Simply we can say xeroxing of the animals. Xeroxing means in the xerox a paper is duplicated, isn't? As the same thing in the cloning, in animal cloning means the animal is duplicated. Okay, in the human being the cloning may be done, it is called as human cloning. Okay, so initially Ian Wilmot in 1997 a scientist created a clone of the sheep. Okay, in the sheep they created a cloning. Okay, so the name of the sheep is called as Dolly. Okay, it is a female fin dorsett sheep. Okay, this type of the sheep, it may be it lived, okay, at the period of 1996 to 2003. Okay, so this is the first clone of an adult mammal. Okay, it was produced by this Dolly female, this type of the sheep. It was first produced by the British developmental biologist. Okay, so the name of the scientist Ian Wilmot and their colleagues. Okay, they worked in the Roslin Institute in the Scotland. Okay, they were working in the Scotland Roslin Institute. Okay, so how they created the sheep in the cloning of the sheep. Okay, so what are the methods they had followed? Okay, the first method, these are the procedure. Okay, these are the procedure they had followed for the development of the cloning of the sheep. Is it? Okay, the first step, how they followed. The first one, the mammary gland cells of the female sheep. Okay, so first number one sheep, number one sheep, a female sheep. Okay, in the female sheep, from the female sheep the mammary gland cells are isolated. Okay, mammary gland cells are isolated and are introduced into the culture medium. They are cultured in the culture medium. And the second number of the sheep, from the second sheep, another sheep, the egg is isolated. Okay, the egg is isolated. Okay, what is the procedure going for egg isolation of the egg? It is isolated, then again inside the nucleus. Okay, the nucleus is located inside the egg. Now the nucleus is removed using the micropipette. Okay, so without nucleus means that type of the egg is called as enucleated egg. That means absence of the nucleus. Enucleated means absence of the nucleus. Okay, so two type of the cells are isolated. The first one, mammary gland cell from the sheep number 1, and the second one, enucleated egg, okay, from the sheep number 2. What is the next step? Now the mammary gland cells and the enucleated eggs are fused together. Okay, now they are combined together. So now they are combined and also cultured in the medium. Again they are cultured in the culture medium. It is going to develop the embryos. Okay, so they developed into the embryos. Under the stimulation it undergoes the development of the embryos. Okay, after the development of the embryo, what is the next step? After 6 days the embryo is introduced into the female sheep number 3. It is introduced into the another sheep. Okay, that sheep, this sheep is called as surrogate mother. It is called as surrogate mother. Okay, now a baby sheep is born after 5 months. Okay, this is the method they had followed in the cloning of the Dolly. Okay, so the first sheep produced in the cloning was named as Dolly. Dolly, it is exactly resemble the mother who contributed the nucleus. Okay, the Dolly, it is exactly resemble. It is an identical copy of the mother. Okay, which mother? Who donated the nucleus? Okay, which one donated the nucleus? Okay, it resembles the Dolly. Okay, this is the story of the Dolly. Okay, so the first one, Scottish blackface. Okay, it is a egg donor. The second one is the fin dorsett. It is a nuclear donor. Okay, so from the egg donor, the egg is isolated. Okay, so the egg is isolated. In the egg, there is a nucleus is located inside the egg, isn't it? Okay, now the nucleus is removed. Okay, now the nucleus is removed. This egg is called as enucleated egg, is it? Okay, from the another one, nuclear donor. Nuclear donor, it is a fin Dorset variety. Okay, here the mammary gland cells are isolated from the shape at depth. That is, the mammary gland cells are isolated here. Now the select a single cell, it is fused with the eunucleated egg. Okay, so mammary gland cells combined with the eunucleated egg. Okay, now it undergoes the culture medium. It is cultured in the culture medium. Okay, culture medium, after that stimulation, it will develop into the blastocyst. Okay, undergoes the division normally and developed into the blastocyst. Okay, now the blastocyst, this is the developing embryo. Okay, the developed embryo can be transferred into the another shape. Okay, this shape is called the surrogate mother. Surrogate mother, in the surrogate mother uterus, okay, the blastocyst is placed. Okay, the blastocyst is introduced into the surrogate mother uterus. Okay, after 5 months a dolly was produced. Okay, this is the story of the dolly, how the Iron Wilmot scientists and their colleagues were produced the cloning of the sheep. That is, the name of the sheep is called as dolly. Okay, dolly, it is a animal cloning method. Okay, then next one is the human cloning. Okay, human cloning as the same procedure of the animal cloning. Okay, cloning means identical copies, Xerox copies, same, exactly. Okay, there is no difference, exactly the same copy are produced. Okay, so human cloning means a man is produced similar to the another man, same. Okay, the Xeroxing of the man. Okay, so that is identical. Okay, similar, there is no difference of the human being. Okay, so human cloning also involve the same steps are followed in the dolly sheep. Is it? A body cell from a man or a lady is first isolated. Okay, then it is cultured in the culture medium. Then again another egg is isolated from the ovary of the another lady. Okay, this is the procedure. Okay, this is the protocol for the human cloning. Now the egg is eunucleated by removing the nucleus. Okay, as the same procedure, the eunucleated egg and the cells are fused in the culture medium. Now the egg developed into the embryo. Egg develops into the embryo. The developed embryo is introduced in the uterus of the surrogate mother. Okay, so the newborn baby is born after 260 days after the gestation periods. Okay, the baby will resemble the parent who contributed the nucleus. Okay, this is the important point here. We have to note down the parent, which parent. Okay, who won, it is resemble means the baby resemble the parent who contributed the nucleus. Okay, nucleus parent, it resemble to the nucleus parent. Okay, it is a human cloning. Okay, in this picture we can understand the human cloning. Okay, this is the human egg donate. Okay, here it is a egg donor. Okay, egg donor here donate the eggs, but in the donate egg the nucleus is removed. Okay, so it is called as eunucleated egg. Okay, from the another person. Okay, this is the first person, this is the second person. From the another person the cells are removed. Okay, the cells are removed. Now the cells contain the nucleus, isn't it? Now the cells and the eunucleated eggs are combined together. Okay, fuse the eunucleated egg and the cell. Okay, it undergoes the stimulation, electricity. Okay, then the fusion. Okay, it will develop into the embryo. Okay, the blastocyst is developed. Now the developing embryo is introduced into the another mother. Okay, this person is called a surrogate mother. Okay, surrogate mother. Now the surrogate mother will produce the newborn baby. Okay, so the newborn baby, newborn baby resembles the egg donated mother. Okay, egg donated mother. Okay, this is the human cloning method. The newborn baby resemble the exact copies of the egg donated mother. Okay, so egg donated mother. Okay, this is the human cloning method. Okay, human cloning and the sheep cloning, the procedure and method is the same for the cloning method. Okay, in the cloning method we can get the same, okay, same person, similar persons. Okay, so what are the advantages and disadvantages of the human cloning we can see. Okay, the first one, infertile couples can produce the children. Okay, so infertility, now the infertility is the common one. Okay, so nowadays it is a major problem, infertility. Okay, so the infertile couples, okay, they can produce the children by the human cloning method. method. And the second one, the parents with the genetical defects can produce the healthy children by cloning method. Okay, nowadays another problem is most of the peoples are affected by various genetic disorders, isn't it? Okay, so that's such type of the parents, okay, they would like to have a healthy children. Okay, so that's why they can follow the human cloning method. Okay, if they are undergoing the human cloning method, they can get the healthy children. Okay, healthy children. Another one, by cloning humans, human organs can be produced for transplantation. Okay, transplantation is the another important area. Okay, it is very useful method for the many people nowadays, is it? Okay, so human organs can be produced by the cloning methods. Okay, this is the another advantage. Okay, finally, the human clone can be treated as the experimental animals, isn't it? The cloned humans, they are treated as the experimental animals. So what are the disadvantages of the human cloning? Okay, so some advantages and disadvantages are always there. Okay, so what are the disadvantages? Mean, to produce a successful clone, many human embryos are sacrificed. Okay, so everyone need a successful one. Everyone needs a better. Okay, better. Okay, that's why the human embryos are sacrificed for the human cloning method. Okay, so the second disadvantage, it may be arise the parental disputes. Okay, parental disputes may be arise. Okay, a problem. Okay, so the law should be changed. Okay, based upon that, the law should be changed upon based upon the human cloning. Okay, then finally, theologists believe that human cloning is against God's will. Okay, it is the real one, it is the real fact, isn't it? Okay, so human creation is the God, human creation only by the God. Okay, so that's why the theologists, okay, they rejected the human cloning method. Okay, so they believe that human cloning is against God's will. Okay, so there are the advantages and the disadvantages of the human cloning method.